Welcome to the video tutorial about how to extract structural insights using 3-decision, based on the CLK selectivity use case.
In our previous video, we showed how to easily generate a dataset containing the relevant structures for our use case. In this video, we are going to compare the structures that we collected to understand structural basis of the selectivity among the CLK family. We can restart the use case from the collection of structures stored in our project. To visualize and examine the differences between structures in a 3-decision project, we need to set a reference structure. In this way, all the data in the project will be pre-aligned to this reference. In 3-decision, the superposition is pocket-based, so you will need to set also a reference pocket. You can exploit the interactive Pocket Explorer to decide which will be the best one to choose as a reference. Then, we can open our project in the 3-decision workspace. Here you have a 3D viewer for the visualization of protein structures. When a protein is opened in the 3D viewer, a set of protein-ligand contacts are automatically displayed. You can also choose which interaction type to show from the full interaction list. In this way, you can easily inspect how ligand binds to the protein, without the need to export the structure in a different program. In our reference structure, we can easily see that the ligand binds to CLK1 with a non-canonical interaction pattern. It interacts with the hinge region through halogen bonds, instead of the classical hydrogen bonding network of ATP-mimicking inhibitors. To compare the binding mode observed with the other structures in our collection, we can superpose them. In a single click, 3-decision allows the on-the-fly superposition of proteins. We can immediately see that in the orange structure, the ligand has a flipped orientation, compared to the other two. We can get more information about this outlier structure from the information panel, accessible from the I button. The orange structure is the wild-type CLK3 structure. If we compare it with the other CLK3 structure in our collection, the white one, we read that this one carries a mutation at the alanine in position 319. We can have a closer look at this position. To better visualize the differences in the binding pocket, we can use the highlight mode. This option in the 3D viewer only shows the differences between the structures above a certain threshold that can be customized. If we focus on the mutation point, we see that the blue structure, which is CLK1, and the mutant CLK3, the one in white, both carry a valine at this position, while the wild-type CLK3, that is in orange, has an alanine. This indicates that the presence of the bulky residue at this location of the binding site induces the different binding pose observed and, consequently, explains the selectivity difference. With 3-decision, we could easily and rapidly identify the structural differences between the structures in our dataset and rationalize the structural basis of the selectivity. Thanks to these structural insights, we have a starting point for designing ligands for overcoming selectivity issues. Thank you for watching. If you want to learn more about 3-decision, visit our website and do not hesitate to contact us.
In the next videos, we will show use cases to illustrate more advanced features of 3-decision.